Okay, today's lecture is going to be on plasma membranes or cell membranes. When you look at a cell, there's all these organelles. There's the nucleus, there's mitochondria, there's the Golgi apparatus, and it's all kind of wrapped up in this membrane. And the membrane doesn't get a lot of love. I mean, it's kind of boring, right? It just kind of sits there. And that couldn't be anything further from the truth. The membrane is extremely dynamic. It's very, very active in the life of a cell. And all too often, students will look at a textbook picture or a model and kind of get the idea that it's just kind of a wall that the cell is in, and it doesn't do anything. It's, like I said, really, really dynamically active. When we look at our plasma membrane, it's made out of what are called phospholipids. These are triglycerides, basically a molecule of glycerol, with three fatty acid tails on the end. Only we've snipped off one of the fatty acid tails, so it's now a diglyceride, and we're going to put a phosphorus group on there instead. And that gives us something really, really interesting. That phosphorus head is polar. It has a positively charged area and a negatively charged area, whereas the fatty acid tails of the molecule are very much not charged. And so now we have something that has a polar end and it has a nonpolar end. This is really interesting. Polar and nonpolar things really don't hang out together. They don't play nice with each other. An example is oil. Oil tends to be made out of triglycerides, and it's very, very nonpolar. It's what we call hydrophobic. In other words, it's afraid of water. It doesn't get along with it. When we look at something that has a charge, say salt, which is sodium ions and chloride ions, the charge gives it polarity. It gives it a direction. We can point at it and go, yeah, this is the positive end and this is the negative end, whereas we can't do that with the fat molecule. Water is the same way. Water has a positive side and a negative side to it, so water is polar. And as I mentioned, the polar and the nonpolar things don't mix. So if you add water to oil, they don't like to play nice with each other, right? They stay separate. On the other hand, if you take a polar substance, such as the salt, and add it to water, all the polar things like to play nice to each other, and the salt will dissolve into the water. Same thing happens with nonpolar molecules. If we have like a pat of butter and we put it into oil and let it warm up, the butter will just dissolve into the oil. So like will dissolve like, but we don't have polar things and nonpolar things mix. On the other hand, we've got these phospholipids, and we've got one end that's polar and one end that's not polar. And this is really interesting because these will rotate so that the polar side of the molecule will face polar water molecules, and the hydrophobic side of the molecule, where the fatty acids are, the nonpolar side, will rotate away from water. What this does is if we use two layers of this type of structure, we get kind of an outer layer where we've got these little phosphorus heads, and we've got our fatty acid tails, and then we've got fatty acid tails and the phosphorus heads of the other group. And if we have water up in here and water down in here, then that hydrophobic region where the tails are doesn't allow water to pass through. So it becomes, in effect, a wall. It separates water. And this is great because that allows the cell to separate water inside the cell from water outside the cell. Now, cells do need to move things in and out, and some of those things are nonpolar and some of those things are polar. So the plasma membrane is really effective, especially for polar substances. It really doesn't let water pass through. Just like you probably have a house or an apartment, and if you try and walk through the wall, it doesn't work too well. You can't pass through it. Instead, you have to use a doorway. So sometimes we can do that. We can have a protein as a doorway, and that allows things that are polar kind of a safe passage through that hydrophobic region of the cell membrane. So to get through the cell membrane, things have to be fairly small, and things that are nonpolar, that are hydrophobic, can easily pass through that hydrophobic barrier inside the plasma membrane. And they really don't need a door. They just kind of like a ghost, I guess, can just pass through the wall. Now, when we look at different lipids in the body, we've got our phospholipids, which I've talked about a little bit, cholesterol, and glycolipids. So phospholipids, as I kind of alluded to earlier, are kind of a little balloon with two tails. So we have our phosphorus head, and there's a little positive charge on one part of it, and there's a little negative charge on the other. And then we've got these two fatty acid tails that are hydrophobic. They don't like water. They're non-polar. Cholesterol, on the other hand, is four interlocking rings of 17 carbon atoms. Sometimes they're called the rings of 17, which is kind of a catchy little phrase. But cholesterol is interesting in that if we add different things onto the outside of it, it becomes different molecules for us. And in the plasma membrane, we use cholesterol, the kind of the vanilla flavor, to help strengthen the membrane. It lends it stability. So all of your cells have cholesterol in them. So cholesterol is not necessarily bad. It is bad if you have too much of it moving around in your blood inappropriately, but we absolutely need cholesterol to stabilize our cells. And then the third type of lipid we have are glycolipids. Glycolipids are fats with sugar molecules attached to one end of it. So that's the glyco part. And that allows for lots of fun things to do on the surface of the cell. They can act as signaling molecules, they can act as recognition molecules and docking molecules, and all those are really critical to the function of the cell. So here's a nice picture of a plasma membrane, and as I mentioned, it looks fairly static. I mean, it's just kind of sitting there. I'll give you a link a little bit later where you can watch a little movie, a little animation that gives you a much better idea of what it's like. All of these little tiny phospholipids, like there's one right here with the little tails. They're all kind of squishing and moving around, kind of like a huge mosh pit. So it's a big dance party going on with the cell membrane, very, very dynamic. Here we can see a protein. You see there's a little, there's a little channel straight down through the middle here that allows polar molecules, like small ions, to pass through. It gets to bypass that hydrophobic region in the center of our phospholipid bilayer. Here's our hydrophobic region again. So proteins that pierce all the way through the membrane are called integral proteins. And then some of the proteins you'll notice, like this little guy down here, are what we call peripheral proteins. They're either only on the inside or only on the outside. So they're on the periphery of the phospholipid bilayer. And then lastly, here's a protein, and then here is a sugar molecule hanging off of it. Those proteins are about half the plasma membrane by weight, so we have a lot of them in every plasma membrane. They float and they move around kind of like big old fat islands in the ocean, and they perform most of the functions of the cell membrane. They allow for transport. As I mentioned, there's the integral and the peripheral proteins. The integral are embedded within and extend across the phospholipid bilayer. They typically have a hydrophobic region in the middle that helps to anchor it into that hydrophobic region through the middle of our phospholipid bilayer. And then the hydrophilic or water-loving regions are on the inside and outside of the cell membrane. And then the peripheral proteins are not embedded in the lipid bilayer. They're just kind of stuck on the outside. So we can use membrane proteins for all kinds of groovy things. We can use them for transport, what allows us to move things both into and out of the cell. Some different ways we can do that we'll talk about later are channel proteins, there's carrier proteins, pumps, there's symporters, there's antiporters, and these are all ways to move things back and forth across the cell membrane. There's cell surface receptors. These bind molecules, these are called ligands. So some of those proteins help to kind of capture different molecules and that allows us to do all kinds of things. For instance, there's a class of chemical called a hormone that you've probably heard of floating around in the blood. And when that chemical attaches to a protein ligand or receptor on a cell membrane, it can cause changes to happen inside of the cell. We can also use the proteins as identity markers. Part of our immune system functions by being able to tell the difference between self and non-self. You can almost think of it as every cell in your body has a little name tag on it that identifies itself. Membrane proteins also function as enzymes. Enzymes are really important. They allow us to do all the chemistry that we do. And they're typically embedded in cell membranes, and that allows us to have kind of an easier place to find them rather than floating randomly inside of the cell. We can use proteins as anchor sites. Perhaps you've wondered why, when you get up in the morning, you don't fall apart into a big puddle of pink goo on the floor. And if not, you should start thinking about things like that. One of the reasons is that these proteins embedded within the cell membranes attach to each other, so one cell will adhere or stick to its neighbor and hold together. It's important. So here's some of our different plasma proteins and what they're used for. We've got transport proteins. Here's a ligand receptor over here with a signaling molecule attached to it. Here's a glycoprotein used as an identity marker. And then enzymes can also be used as, proteins can also be used as enzymes, for example. So here's the molecule it's going to act on, called the substrate. Here is the active site, and then after it runs into the enzyme will then convert it to a product. Here's a peripheral protein, this little guy right here, stuck to an integral protein, and we're using it to anchor some different structures inside of the cell. And then the last little picture here shows us two different plasma proteins. So this is cell number 1, and this is cell number 2, and they're held together with these docking proteins, these adhesion proteins.